In the last practical, we had discussed about the morphology and microscopy of the clove. So today we'll be discussing the morphology, microscopy of the Senna, as well as we'll be discussing about the stomata, its classification and different types of stomata. So this will be our aim of today's practical, to identify the given food drug by morphology, microscopy, microchemical test, and chemical test, if any. So this microchemical test, these are nothing but these are the staining. You have to stain your slides. So this all the staining procedures, they are included under the microchemical test. Most widely used staining reagent, they are the fluoroglycerol concentrated HCl, then ruthenium red, Sudan red 3, so Senna belongs to which class of the drug? Can anyone?
Anthracene glycoside.
Anthraquinone glycoside.
Yes, it is an anthraquinone glycosides. Correct. So just we'll start. As I told you, first before going to the details of the morphology and microscopy of the Senna, we will first discuss about the stomata, right? So what do you mean by stomata? Can anybody? Okay, this stomata, they are the diagnostic features of the plant. What do you understand by the diagnostic features? The stomata, trichomes, they are the diagnostic features. So what do you understand by this term, diagnostic features? Can anyone?
Identification marks.
Correct. So diagnostic feature means the structure, the parts by which the plant, it can be differentiated from the other ones. Okay. So this many of the plants, they are being distinguished on the basis of their stomata, presence of their stomata and presence of their trichomes. Okay. So today we'll discuss about the stomata, its classification, and what are the different types of stomata. So basically this stomata, it is made up of two kidney shaped guard cells. Okay. So this kidney shaped structure, they are called as the guard cell, and they are embedded in this subsidiary cells. Okay. They are attached to the subsidiary cell. So the subsidiary cells are nothing but these are the same epidermal cell. In the plant, we know that there are epidermis, that is a dermal tissue system. So this epidermal cell, what will happen? Same epidermal cell, they grow into size and shape, and they attach to this guard cell, and they are termed as the subsidiary cells. Okay. So these subsidiary cells are nothing but these are the same epidermal cells. Only the difference between the epidermal cell and subsidiary cell is that this epidermal cell, they get elongated in their shape. Okay. If you see size and shape, it is always bigger than this epidermal cell. So stomata, it is nothing but it is a combination of these two kidney shaped guard cell which are attached with the subsidiary cell, and they are having a pore in the center. Pore means it is called, it is nothing but it is an opening. Okay. So this opening, it is called as the stoma. Okay. So what is stomata? It is made up of two kidney shaped or bean shaped guard cells which is attached to the subsidiary cells, and it is having a opening into the center. That opening, it is called as a stoma. Okay. So you got the definition of the stomata? Any doubt, confusion?
No, sir.
So,
Okay. So what is the function of this stomata?
so transpiration or exchange of gases.
Exchange of gases. So normally the stomata, they are present at the downside. Okay, when those plants, the terrestrial plants, in that the stomata, they are present at the lower layer of the leaf, whereas in aquatic plants they are present at the upper surface. Okay, and as correctly said, the function of the stomata is the exchange of gases. Okay, so the carbon dioxide intake and expulsion of the oxygen, it takes place through this stomata. Okay, so all these stomata, they are present at the in the epidermis part, dermal tissue system. Okay, there are three main tissue system: dermal tissue system, then fundamental tissue system, and vascular tissue system. Dermal tissue system, it is composed of the epidermis. Fundamental tissue system, it is composed of the parenchyma, collenchyma, and sclerenchyma. And vascular tissue system, it is composed of the xylem and phloem. So these stomata and trichomes, they are a part of the dermal tissue system. Okay, so always they are present at the uppermost part. Okay, and in the leaves they are present in the lower side, not at from the upper end. So this was about the brief introduction to the stomata, its definition and its function. Okay, now we will see the classification of the stomata. So on the basis of this guard cells, okay, what are the guard cells? Can anybody? Just now we discussed guard cells matlab kya hai?
So that kidney shaped cells.
Correct. Okay, so classification, it is based upon two types. First classification, it is based upon the guard cell, that is the kidney shaped cells, and second, it is the subsidiary cell. Subsidiary cell matlab kaun se? Come on. Subsidiary
So
cells.
around the guard cells.
Yes, those bigger cells which are attached to the guard cell, they are called the subsidiary cell. Okay, and basically subsidiary cell, they are nothing but they are the epidermal cell which are getting elongated in size and shape. Okay, so mainly stomata, they are classified on the basis of the type of guard cell which are present and the subsidiary cell. Okay, so first we'll see the classification which is based upon the guard cell, that is the two kidney shaped guard cell. Okay, so on the basis of the guard cell, stomata are classified into four types. First one, it is a moss type stomata, gymnospermous, gramineous, and dicotyledonous. Okay, so in the moss type, here the guard cells, they are the united one, they are stick to each other, whereas in the gymnospermous, these guard cells, they are of oval shape. Oval shape matlab elongated, egg like. So in the moss type, these guard cells, they are united one. In the gymnospermous, these guard cells, they are oval shape or egg like shape. In the gramineous, these guard cells, they are of the dumbbell shape. Okay, and in the dicotyledonous, the guard cell, they are of the bean shaped or kidney shape. Normally most widely appearing stomata, it is of the guard cell, that is the kidney shape or bean shape. So on the basis of the guard cell, there are four types of stomata. Moss type stomata, where the guard cell, they are united one. Gymnospermous, where the guard cell, they are oval shape. Gramineous, dumbbell shape. And dicotyledonous, that is the guard cell, they are kidney or bean shape. So this was about the first type of classification, which is based upon the guard cells. Have you got this? Any doubt, confusion regarding this first classification?
Yes, sir.
Okay. Now we'll coming to the subsidiary cell. This is very, very important, and mostly the distinguishing characters are being done on the basis of this subsidiary cell. Okay, so this classification, it is very, very important. So on the basis of this subsidiary cell is nothing but these are the epidermal cell which are got elongated into size and shape, and they are attached to this guard cell. Okay, so there are 5 different types of stomata according to this type of subsidiary cell. Okay, so first one, it is parasitic or rubeosis stomata, diastatic or caryophyllous stomata, anisocytic or cruciferous stomata, anomocytic or ranunculus stomata, and actinocytic stomata. Please remember, you have to by heart the names. Okay, and both the common name as well as the scientific name. Okay, in many times for the GPAT examinations, the question, it may be asked on the basis of this scientific name, that is rubiaceous. So question it may be asked, what give the chemical constituents of a drug which is having the rubiaceous type of stomata, belong to the family Leguminosae or belong to the category purgative. Okay, at that time you have to give the details of the Senna. Okay, so in many of the time the scientific names are being asked as a question in the your GPAT examination. So it is very, very important to remember both this name, okay, common name as well as the scientific name. Okay, so on the basis of the subsidiary cell, there are 5 different types of stomata. First is parasitic stomata, second is diacitic stomata, third is anisocytic stomata, fourth is anomocytic stomata, and fifth it is the actinocytic stomata. So first there is the parasitic stomata. Here the long axis of this subsidiary cell, they will be always parallel to the stoma. Okay, stoma means, what is stoma?
So the hole present in the guard cell.
Exactly. Okay, so here this long axis of this subsidiary cell, they will be always be parallel to the stoma. Many of the times students, when the question is asked in the synopsis, they write long. If the questions are what is the parasitic stomata, in the hurry what they are writing, long axis of 2 subsidiary cells, they are parallel to the stomata. If you are writing the stomata, your answer will be wrong. Okay, ये किसको parallel है? Parallel there to the stoma and not the stomata. So you should be very, very careful while writing the answer. Okay, so parasitic stomata means here the long axis of subsidiary cell, they will be always parallel to the stoma. Stoma in nothing, it is a pore, it is an opening through which the gaseous exchange is taking place. So in the parasitic stomata, the long axis of subsidiary cell, it will be always parallel to this stoma. Example, it is Senna and Coca. Diacitic stomata or caryophyllus. What is diacitic? Here the long axis of subsidiary, they will be always perpendicular to the stoma, right angle to the stoma or perpendicular to the stoma. In the parasitic, they will be parallel to the stoma, whereas in diacitic, they will be perpendicular or right angle to the stoma. That is the opening. So this was about the diacitic stomata. Now the anisocytic stomata. So in this, what happened? The stomata, it is surrounded by the 3 subsidiary cells. Okay, and one of the subsidiary cell, it will be always smaller than the other. Okay, so see here 1, 2, 3. These 2 subsidiary cells, they are bigger than the one. Okay, so in the anisocytic stomata, what is the condition? Always there will be 3 subsidiary cells, and one subsidiary cell, it will be always smaller than other 2. This is called as the anisocytic stomata. Okay, now anomocytic stomata, here all subsidiary cell, they are irregular, they will be not matching. Okay, whereas in anisocytic, 2 are similar in size, but one is always smaller, whereas in anomocytic stomata, all the subsidiary cell, they are unequal size. Okay, and number is also not necessary, it should be 3, it may be either 2, 3, it differ according to the plant variety. Whereas in anisocytic one, always there will be 3 subsidiary cells, and one will be always smaller than the other 2. Whereas in anomocytic stomata, there are irregular cells. And last one, it is the actinocytic stomata or radiated cell. So radiation means they are arranged in a radial fashion, or they are arranged in a circular fashion. These are the subsidiary cell which are arranged in a circular fashion. So this is called as the actinocytic stomata. So these are the 5 different types of stomata which are based upon the arrangement or type of the subsidiary cells. So first classification was based upon the guard cell, and second classification, it is based upon the subsidiary cells. Parasitic stomata, wherein the long axis of subsidiary is parallel to the stoma. Diacitic, the long axis of subsidiary cell, they are perpendicular right angle to the stoma. Anisocytic means always there will be 3 subsidiary cells, and one will be always smaller than the other 2. Anomocytic stomata, irregularly celled, and actinocytic stomata means subsidiary cells, they are arranged in a circular or radial fashion. You got this classification of the stomata and description of the stomata, its function. Any doubt, confusion?
No, sir.
So shall we move ahead? Now we'll be discussing about the morphology and microscopy of the Senna leaf. Okay. So this you can take down afterwards. First we will see a video regarding the description of this morphology, microscopy and chemical test of the Senna leaflet. Today we will be discussing the study of morphology and microscopy of the Senna leaflet. We all know that Senna belongs to the class of anthraquinone glyphosate. Biological source of the Senna is dried leaflet of Asia angustifolia or belonging family legumes. When we say leaflet means leaflets, they are arranged in the pairs to a single lamina. 2 pairs or 3 pairs or 4 pairs are arranged, whereas a single leaf means to a lamina, a single leaflet is there. So basically Senna it is a leaflet. Coming to the morphology, the length of the single pair, a single leaf, it is around 2 to 5 centimeter. It is having an apex of 7 to 8 millimeter. When we look at the venation, so Senna is having the reticulate venation. That means these veins they are protruding towards the margin. Okay. And this is the base of the Senna leaf. Now, the microscopy of the Senna, it is divided into 3 types, 3 zones. First, it is the upper epidermis. Then this one it is the mesophyll, and this one it is the lower epidermis. The microscopical structure of the Senna, it is a isobilateral one. When we say isobilateral means there are 2 layers of the palaceid cells. So these are the palaceid cells. This is the first layer of the palaceid cell, and this is the second layer of the palaceid cell. So when a leaf contains 2 layers of the palaceid cells, which are present at the upper epidermis also, as well as the lower epidermis also, they are termed as the isobilateral. Whereas if a single layer is present, then that leaf is termed as the dorsiventral. So Senna basically it is a isobilateral nature. The first layer, that is upper epidermis, as usual, it is composed of the thin layer cell, living cells, having the function of the protection. Below this, there are the layers of the palaceid cells and the parenchymatous cells. The mesophyll, it is composed of the vascular bundle, which are of xylem and phloem. Also, there are crystal sheets in the parenchymatous cells, which are present in the mesophyll layer. Then this lower epidermis, it is having the layers of parenchymatous cell, and the structure, which are the comma shaped structure, which are protruding from the epidermis. These are nothing. These are the trichomes, and the trichomes which are present in the Senna leaf, they are of the covering trichome. So this one about the microscopy and morphology of the Senna. Now we will see the section cutting of the Senna leaf. So this is Senna leaf, dried leaflet of the Senna. These are the boiled one. First we have to take the leaf, place it on the slide. Now, to have a good section, to ensure a good section, always we have to take your TS from the midrib portion. Okay, so this is the midrib portion. You have to discard the apex one, as well as you have to discard this. So this is again the midrib portion. The reason behind to take the section from the midrib portion is that in the midrib portion the diameter is large. That's That's why all the microscopical features will be distinctly visible, or they will be clearly visible. Okay. So now first, these are the boiled senna leaves. Now, as I told you, first we have to ensure that we have to take the section from the midrib region. Again, in the last practical, I told you, you should always use a fresh blade. Okay. So first thing, you have to ensure the midrib portion. So what I am doing, I am cutting this base first. And again, I am cutting this. Okay. So what I will do now, I will just discard this. Now I have to take the section from this midrib region. So there are two ways of taking the section of the leaf part. First is you have to place the leaf on the screen side. Then, as usual, place your finger and take the thin section as much you can. Okay. This is for the first. These are the sections. These are the thin sections which are taken, and now we will selecting from this one. Then second one. Second method is by using the pith method. So in the pith method, what has to be done? There is a small piece of either potato. It is being cut, and this leaf, it is placed in between this potato. Consider this as a piece of potato. So this leaf, they are placed in this potato, and then the sections, they are taken in this fashion. These are the two ways. Whatever the method which is feasible to you, you can take. Okay. So these are the sections, fine thin sections of the senna leaf. And now in the last practical, I showed you the mounting technique. Same way, you have to take the section, mount it on the side, and see it on the microscope. So this was about the section cutting of the senna leaf. Now, as we all know that senna, it is belonging to the class of anthraquinone glycoside, and we all know that for the anthraquinone glycoside, Braun Trigger test has to be conducted. Okay, to confirm the presence of anthraquinone glycoside or anthracene glycoside, Braun Trigger test has to be done. So now we will see the Braun Trigger test. So in the Braun Trigger test, first what we have to do, you have to take the senna leaves, then you have to add dilute HCl, and you have to boil the leaves in this dilute hydrochloric acid. So what will happen? This will give the hydrolysis. Okay. After that, you have to filter this solution, and to the filtrate, you have to add benzene. When you add benzene, you will be having such two layers. Okay, I am repeating once again. First, you have to take the senna leaves, you have to boil in the dilute hydrochloric acid. After boiling, you have to filter it, and to the filtrate, you have to add benzene solution. So when you add benzene solution, you will be having these two layers, first layer and second. Now we have to separate this organic layer, that is upper one. So now in the test tube, I am separating this organic layer. So now I have separated the organic layer. Now in this organic layer, that is benzene layer, now we have to add the strong ammonia. This is ammonia solution. add this strong ammonia solution. Now you have to shake it. Upon shaking, you will see that this ammoniacal layer, it turns pink. So this pink color, it is confirming the presence of anthraquinoglycogen. After standing for a few minutes, this pink color, it will be clearly visible. You have to stand for a few minutes. So this pink color, it is confirming the presence of anthraquinoglycogen. This is the modified brown triangle test. So today we have discussed about the morphology, microscopy, and chemical test for the Senna leaf. So was the video clear to you?
Yes, sir.
Sir.
Yes, there was a slight correction. The test which I explained, it is a Braun-Trager's test. Okay, there is a difference between Braun-Trager's and modified Braun-Trager's test. At the end of the chemical test, I had uttered modified Braun-Trager's test. Okay, actually it was not a modified Braun-Trager test. It was only a Braun-Trager's test. Okay. Now we will see what is the difference between the Braun-Trager test and modified Braun-Trager test. See, for the Braun-Trager, that was the first one. We had taken the extract, we had added dilute H2SO4, filter it. To the filtrate, we added the benzene, separated the organic layer. In the organic layer, we added the ammonia, and we got the pink color. This is the Braun-Trager test. Whereas, what is the difference in modified Braun-Trager test? Only this, that is the addition of FeCl3. It is the difference. Okay, so the demonstration which was shown to it was for the Braun-Trager test, but at the end of that video, by mistake, I told you the modified Braun-Trager test. Okay, so that is a correction in this. So only difference between the modified Braun-Trager test and Braun-Trager test, it is the addition of 5 ml of FeCl3 in the modified one. Whereas in the Braun-Trager test, FeCl3 it is not added. So this was about the chemical test for the Senna. So you got this practical, any doubt, confusion?
No, sir.
Okay. Then in the video, we had not discussed about the organoliptic character. Okay. So organoliptic character, normally the Senna, it is having the pale green color, odor very light, and taste it is bitter. Okay. Always if you have a bitter taste of the Senna powder, and the category or main use of the Senna, it is a purgative drug, anti-purgative. So now you do one thing, you just take down the stomata and this classification and this microscopy. Okay. Yes, first draw the diagram of the stomata, and this stomatal pore, you can label it as a stoma. Okay, in the image it is given as a stomatal pore, but for better understanding you can label it as a stoma. That is the opening, it is called as the stoma. And definition of stomata, it is the structure which are made up stomata, they are made up of two kidney shaped guard cell surrounded by the subsidiary cells having a pore or opening in the center, which is called as the stoma.